So welcome back, class. We are in week 10 of semester. Oh, hello there. We're in week 10 of semester, and tonight we'll be looking at 1.5 lectures we'll be covering tonight. And this particular lecture is really all about plant anatomy, fundamentals of plant anatomy. So we'll look a little bit more at plant tissues, the major tissue types, and some characteristic cells. And then we'll go into a look at the anatomy of stems, roots, and leaves. And of course, those of you that will go on in second year to study the subject of plant physiology will go into a lot more detail. And some of these lectures are very important, sort of basic, give you the basic information that you need going into that subject. So let's start with plant cell walls. And as you all know, most plant cells are surrounded by cell walls, and cell walls function primarily to define the size and shape of cells, but they do also contribute to metabolic processes. There are some enzymes that are located on the plant cell wall, and they're also thought to be involved in communication between cells. So the plant cell wall really functions as an extracellular matrix, you know, similar in a way to what we find in between animal cells, but of course it's a lot more rigid than your typical animal extracellular matrix. And lying in between adjacent cells is a region called the middle lamella, which is made up of a complex polysaccharide called pectin. And this pectin binds cells together. All right, so let's take a look at a few parts of a cell, plant cell. So we've got a few cells around here, and then this one in the center has been lifted up above the plane of the other cells. So we can see to the outside of the cell in purple, what's called the primary cell wall. I'll come back to that in just a minute. Just to the inside of that, this thin brown region, of course, is the plasma membrane. And then to the inside, we've got that large central vacuole within the cytosol, the nucleus, and of course, you know, there's lots of other organelles and structures within the cytosol that's not shown here. Okay, in between adjacent cells, so this thin purple region here, this is called the middle lamella, that pectin-rich middle lamella. Okay, so plant cells are surrounded by cell walls. Most plant cells, remember there's always exceptions. Most plant cells are surrounded by a cell wall, and that cell wall is called a primary cell wall. And later on in the development of plant cells, some of them also develop an additional wall called a secondary cell wall. And I'll come back to that in just a minute. So the primary cell wall is the first cell wall that's actually formed when a cell first develops. It's quite thin, and it's very flexible. It's very hydrated, highly hydrated, and it can easily expand as the cell is still growing. The primary cell wall, as you know, is composed primarily of that structural polysaccharide called cellulose. And you might also remember that cellulose is made up of repeating units of glucose. Cellulose forms microfibrils, and together with various pectin molecules and non-cellulosic polysaccharides and various proteins. That's basically the composition of the primary cell wall. And as I said, that first cell wall that develops, the primary cell wall, is quite flexible, so it's able to extend as the cell grows and matures. All right, but primary cell walls actually vary in thickness. There are thick regions, there are thinner regions. Those thinner regions are referred to as primary pit fields. And the reason that they are thin in comparison to the rest of the cell wall is because this is where we find numerous little channels, fine channels called plasmodesmata, which you were introduced to last week. And these plasmodesmata connect cell to cell. Okay, and we can see that here. So we've got one central cell here and then a portion, or rather several portions of adjacent cells shown around the outside. So we can see around here, this is a thicker region of the cell wall, of this central cell, but then as we get to this region here, you can see that it's quite thin, and that's the region called the primary pit field. And you might also be able to see some fine lines there, and they are the plasmodesmata that connect each cell to the neighbouring cells. But as some cells mature, they might lay down additional cell wall material to the inner surface of that existing primary cell wall. And the new materials form what's called a secondary wall. And that secondary wall can be much, much thicker than the primary wall itself. The secondary wall is usually formed in 3 distinct layers, each consisting of cellulose microfibrils laid down in different orientations. So if you just imagine a piece of plywood where you've got fibres running in different directions, that's the way that those cellulose microfibrils are laid down. But what's really unique about secondary cell walls is that it's actually strengthened by the addition of a very complex substance called lignin. Lignin is a type of chemical called a polyphenol. And we'll come back to lignin later on. And of course, some of these cells have lots of thinner areas or depressions called primary pits. And for the same reason, because they're the regions where those plasmodesmata are connecting neighbouring cells. Now, let's revisit the topic of plant tissue systems. And you might remember from last week that there are only 3 major tissue systems in plants. And you might also remember that the primary plant body is composed of cells that are produced by 2 meristems. One's called the shoot apical meristem, or SAM, and the other major meristem is called the root apical meristem, or simply RAM. All right, our first tissue type is the dermal tissue. And the major type of dermal cell is the epidermis, epidermal cells. These are normally cells on the surface of the plant, the surface of the plant body, that is the shoot and the actual roots itself. So those surface cells, normally 1 layer, sometimes 2 layers of epidermal cells. And these are the cells that segregate the internal plant tissues from the external environment. Okay, so major type of dermal cells. Epidermal cell structure varies depending on where it's found, whether it's in the stem, root or leaf, although its main function is in protection and facilitating exchange of materials. Now, there are some very unusual types of dermal tissues, and this includes the endodermis and the exodermis. And these are dermal tissues that are not found on the surface of the plant, but rather within the plant itself. And we'll come back to those later on. All right, our next major plant tissue system is ground tissue. Most of the plant consists of ground tissues, and there's lots of different types of ground tissues. These tissue systems include parenchyma, collenchyma, sclerenchyma, and these make up the bulk of tissues in stems, roots, and leaves. And we'll look at some examples of these. So here's one type of parenchyma cells. These are storage cells of potato tubers. And you can see that each of these cells contains lots of starch granules, which you would expect in cells of potato tubers. And the starch granules have been stained black by using iodine. So that's one type of parenchyma cell. Here's another type of parenchyma cell called erenchyma. And erenchyma cells have these very large air spaces, which enables a better movement of gases in these particular cells. All right, another type of cell, these are transfer cells. And these cells, you probably can't see it very clearly here. Let me enlarge this a little bit. Okay, that's better. So these transfer cells, they're found in an embryo of a seagrass. And what they have here, where it looks like there's little lines in between each of these cells, those little lines are actually ingrowths of the cell wall. And basically what that does is increases the surface area of the plasma membrane, and it allows more efficient transfer of materials between the cells of this embryo and the cells of the actual parent plant. So they're transfer cells. Okay, and then we have parenchyma cells, and these are cells that are normally quite large with thin primary walls and a well-defined middle lamella. All right, so we've looked at transfer cells of the last slide. So these are the cells that had those ingrowths of the cell wall that increases cell surface area for rapid transfer of molecules. On the slide before that, we looked at erenchyma cells, and these are spongy cells with large air spaces adapted to low oxygen environments. Storage parenchyma, so these are the cells that we saw in the potato tubers, and of course they function to store nutrient reserves. But here's a very important one, chlorenchyma cells. These are our photosynthetic cells, and they're found in leaves and also often in stems, and also other plant tissues as well, depending on the plant species. Okay, our next type of ground tissue, this is collenchyma tissue. It's primarily supportive tissue which provides strength to plant parts, but not just strength, but also flexibility. So if you imagine the stalk of a leaf, that's where you would find this type of tissue. So the cells tend to be elongated, they have thickened cell walls that contain extracellulose. Now, a similar type of ground tissue is sclerenchyma tissue. And this is also supportive tissue, but it imparts rigidity and strength rather than flexibility and strength. Two major types of sclerenchyma cells are shown here, fibres and sclerids. But what's interesting about sclerenchyma cells is that once they actually mature, they lose most of their cytosol and their cell contents, and they're actually dead cells at maturity, at functional maturity. They still carry out their task, but they are dead cells at maturity. All right, now moving on to our next tissue type, and this is the all-important vascular tissue. And you might recall from last week that vascular tissues are involved in transporting materials around the plant body. Vascular tissue consists of xylem tissue and phloem tissue, and there are several different cell types found within the xylem and within the phloem. So you might recall that xylem is the vascular tissue that transports water and dissolved minerals from the point of uptake at the plant roots up to the leaves. And the major cell types in the xylem tissue, well, the most prominent cell types are the vessels and tracheids, and we'll look at those on the next slide. The other major type of vascular tissue is the phloem, which transports the products of photosynthesis, in other words, carbohydrates or sugars, primarily from the leaf, which is the major photosynthetic organ, throughout the plant to actively growing areas or to places of storage. So of course, while the xylem transports water and minerals always from the soil solution upwards through the plant, the phloem transports carbohydrates in all directions, in an upwards direction, down, and also laterally. All right, so let's concentrate on the xylem for now. All right, and here is one of the major types of transport cells in the xylem, and these are called vessels. And vessel cells are common in flowering plants. All right, let's look at a vessel here. So this is a young vessel element. Let me make that a little bit smaller so we can capture everything. Okay, a little bit smaller again. Okay, there we go. All right, so to the left, we have one vessel element here, one complete vessel element here, and then this lower part is a portion of another vessel element. And even though vessel elements, when they're first formed, they're actually living cells, but once again, when they mature, they lose all their cell contents. The ends of the cells actually degrade, they degenerate. So what you're left with is basically a hollow tube. So at maturity, vessel cells, or rather they're referred to as vessel elements, they are dead cells once again at functional maturity, but they still function to transport water and dissolved minerals through the plant. Okay, so they have these open end walls and they stack on top of each other, as you can see, to form this pipeline, if you like, through the plant. And that permits this unimpeded flow of water from one vessel element to the next. All right, here we have a large, or rather two large mature vessel elements. And once they get to this point, they've completed the formation of secondary cell walls. So all vessel elements have these thick secondary cell walls that are impregnated with that substance called lignin. And if it wasn't for the lignification of secondary cell walls, then those cell walls would actually collapse under the pressure of the water that's constantly moving through. And those cell walls are actually very, very tough. You can take a small section of a plant stem and place that on a microscope slide. You can dissolve it with some, dissolve the cells with some dilute hydrochloric acid. And what you'll see remaining is the actual secondary cell walls of the xylem. All the other cells will be dissolved, but you'll see these little tubes sitting up on your microscope slide. So very, very tough as a result of that lignification. Okay, to the right here, we have another vessel element. And you can see this arrow representing the upwards movement of water and nutrients. Over here we have another one. But you can also see that vessel elements are very closely associated with living support cells. And these are called xylem parenchyma cells. So that's vessel elements. And here's the other common type of xylem cell. These are called tracheids. And tracheids are common to all vascular plants, not just flowering plants. And they differ from the vessel elements in that they have intact end walls. They're not open at each end like the vessel elements are. And the water actually moves from cell to cell through what are called bordered pits in the cell wall. So you can see one tracheid here, which has been partially opened up, and part of another tracheid there. And these pores that you can see in the actual cell wall, they're called bordered pits. And here the arrows indicate the direction of water movement from one tracheid to the next. Moving on now to the phloem. So the phloem is also part of the vascular system, and it comprises specialised cells called sieve tube cells. And these sieve tube cells are responsible for the movement of the products of photosynthesis through the plant. And they are living cells, but they are associated with helper cells or support cells called companion cells, because sieve tube cells, although they are living cells, they actually do lose a lot of their organelles, so they still need support from other cells. Sieve tube cells are elongated. They have thin primary cell walls composed mostly of cellulose, and the companion cells that are associated with them control the activity of sieve tube cells. And let's have a look at that on the next slide. So here is our typical sieve tube cell. And you can see why it's called a sieve tube cell, because each end resembles a sieve. So the end of the cell is called a sieve plate. But you can also see that in the actual side walls of this cell, there are also numerous tiny pores, which are called sieve areas. Okay, and to the right here, we can see the close association with a companion cell, which provides metabolic support to the sieve cells. All right, so enough about plant tissues, and now let's move on to the major plant organs. But before we do, let's have a look at the structure of a typical plant, which I'm sure everyone's familiar with. And I'll just enlarge that a little bit. Oh, that didn't work. All right, can everyone see that clearly? Or is that hiding? Have I got that hiding behind this chat box? Okay, yep, it's clear. Thank you very much. All right, so let's start firstly. Oh. All right, let's start first with the shoot system. So, of course, the above ground vegetative components of the plant is referred to as the shoot system. All right, so here we have the actual, the tip, the growing tip of the shoot. And who remembers those cells, the meristem cells that are found within the shoot tip? No, cross that out. Shoot apical meristem, the SAM. So that's found just below the actual shoot tip. RAM and SAM, very good. Very easy to remember, aren't they? All right, so that's the location of the shoot apical meristem. Everyone's familiar with leaves, but of course the blade itself is given a name, and that name is the lamina. Lamina. I'll just write that there. Okay, so that's the blade, and the leaf stalk is called the petiole. There it is there, so I don't have to write it. Petiole. All right, of course we have the stems, we have the point of attachment of the leaf to the stem, and this point of attachment is called the node. The point where the leaf attaches to the stem. Here we have the region of stem in between this node and the next node, and that region of stem is called the internode. All right, so even though we have a shoot apical meristem up here within the shoot tip, we also have these branches starting to form, these lateral branches. And when they're starting to form, they're referred to as axillary buds. And axillary buds, of course, are shoots. So they also have their own little shoot apical meristems within the tip. All right. Oh, of course. And we have these veins within the lamina of the leaf. And the veins, as you can imagine, that you can see, and the midrib, the major vein running down the centre, these are all parts of the vascular system, which is visible. Right, so that's the shoot system. Let's now take a look at the below ground part of the plant, the root system. And we'll start firstly with this primary root, often referred to as a taproot. And we'll start down the bottom here. So at the very tip here, this is called the root cap. Come back to that. Just behind the root cap, we have a region here called the root tip. And within the root tip is where you'll find the root apical meristem cells. All right, just behind that, we have some root hairs, not to be confused with lateral roots or root branches that you can see here. All right, within the actual primary root, we can see the beginning of the vascular system, just a central core of vascular tissue. Remember the xylem and the phloem extending up through the root here, and then also branching out into the lateral roots. As that core of vascular tissue hits the stem, it starts to branch out. So you can see it's branched out into 2 points here. Well, actually only 2 branches are shown here. There's actually multiple branches that form within the stem. And then as the vascular tissue reaches the leaf, it branches out into the leaf to form that midrib, and then this whole network of veins, leaf veins. All right, so that's the general structure of a plant, and I'll just clear all that. And then take this back. So now we're going to start with the stems. And before we do, okay, let me just change that again. Before we do, let me just need to look very briefly at this term, dicotyledons. Is anyone familiar with monocots and dicots? Okay, so all flowering plants are divided. Yes, someone's familiar with it. All flowering plants are divided into two groups, are placed into one of two groups. One group is called the dicotyledons, or simply dicots for short, and the other group is referred to as the monocotyledons, or simply monocots. Okay, and there are various characteristics, if you like, that are associated with the two groups. So here we're just looking at the stem of a dicotyledonous plant. All right, so imagine if you took a very thin section of a stem and then basically flipped it over so you could see all the cells within the actual stem. This is most likely what you would see. Well, if you actually stained it, that is, so that you could see these different colours differentiating different cell types. So you would see firstly this outer layer of cells, which is the epidermis, most likely a single layer of cells. I probably should enlarge this too. That. I'll just enlarge this a bit, maybe a bit more. There we go. All right, that should be clearer. All right, so outer layer of cells, the epidermis, and here's the terms here to the right. And then we can see a cluster of cells here and a similar cluster there, and so on. And these clusters of cells that you can see are called vascular bundles. And each vascular bundle, one of them circled here, so they form a ring around the inside of the stem, as you can see here. And that formation of vascular bundles into a ring is typical of most of the dicots. If we were looking at a cross-section of a stem from a monocot, those vascular bundles most likely would just be scattered randomly throughout the stem rather than forming this ring. So that's basically one of the defining characteristics of monocots versus dicots versus monocots. All right, so vascular bundle. What's found within a vascular bundle? In the vascular bundle, you'll find cells of the xylem and the phloem. The phloem is typically located to the outside of the vascular bundle, so closest to the outer epidermis, and then the xylem is typically located to the inside of the vascular bundle. The cells to the inside of the stem, these large cells that you can see, they are referred to as pith cells. And the vascular bundles and the pith together form a central cylinder, which is referred to as the stele. Let me move that to the side so you can see that. Okay, and there is the word there, stele. So the pith cells and the vascular bundles together form the stele. All right, then we have these cells around here in between the ring of vascular bundles and the outer epidermis. And these are collenchyma type cells, which together form what's called the cortex of the stem. Right, let me clear all that. All right, so let's recap. So the vascular tissue often forms a single ring in dicots, and it separates the stem outer cortex from the inner pith cells. The phloem forms outside the xylem on the same radius within those vascular bundles. The earliest forming xylem, which we didn't actually see there, is referred to as the protoxylem. And then outside of this develops the larger, thicker-walled xylem cells, which is called metaxylem. So protoxylem and metaxylem. And yes, we can see that here. So once again, we have a vascular bundle. And this time the cells are not as well differentiated because of the way that it's been stained. But here we have the earlier protoxylem and then the larger thick-walled metaxylem cells. And you can clearly see how thick those cell walls are there compared to the early formed xylem. And then, of course, the phloem here. All right, here we can see a single layer of epidermal cells. And we can also see, well, not very clear, but these cells here in between each of the vascular bundles, these are a type of those sclerenchyma tissue that we talked about earlier. And these particular cells are called fibres, intervesicular fibres. You don't have to remember those names. Okay, so the type of growth that we've been talking about so far, that originates from the shoot apical meristem, the root apical meristem, and so on, it's all referred to as primary growth. And all plants undergo primary growth. But when we're talking about woody type dicots, such as trees and shrubs, and even smaller herbaceous plants can also undergo woody growth, these plants undergo what's called secondary growth. And secondary growth adds girth to the plant. It makes stems much wider. Okay, so secondary growth promotes radial growth. Unlike that primary growth, which originates from the SAM and the RAM, they basically add height to the shoot system or depth to the root system. Okay, so secondary growth basically originates from two different meristems within the plant. And these meristems are referred to as cambiums. The two types are shown here. One's called the vascular cambium, which will give rise to woody tissue. And the other meristem is called the cork cambium, which gives rise to bark. And these two types of meristem produce sheets of cells laterally to add girth to the stem. So let's look at that in a little bit more detail. First, starting to the left here, so obviously what you're seeing is a portion of a woody stem, obviously a trunk, and I'll just enlarge that a little bit. There we go. All right, so let's look at some of the names here. So to the outside of this stem, we have a layer of cork. All right, here, so there's just a line, a ring here, which you can see here. This is one of the cambium, one of the meristems called the vascular cambium. And we're just going to take a small region of that and enlarge it so we can see that over here. All right, so the cambium, the vascular cambium, basically is represented by these three layers of cells in the middle here. Okay, so that's the vascular cambium. So these are undifferentiated meristematic cells. And when you hear that term meristem or meristematic, think of stem cells, which you find in animals. Same sort of thing. All right, they're meristematic cells, undifferentiated, and they will divide to produce secondary phloem to the outside of the vascular cambium and secondary xylem to the inside of the vascular cambium. All right, so imagine that here we have the outside of the stem up here, And let's take this, this cell here, the vascular cambium, as an example. This cell will divide into, in this direction. So this cell here will form a cell towards the outside of the stem, a secondary phloem cell, and this cell here, as a result of that cell division process, remains undifferentiated as part of the vascular cambium. Let's take this cell here now, this vascular cambium cell. It will divide into, this new cell here will form a cell towards the inside of the stem, and it will form a secondary xylem cell. This cell here, this resulting cell here, remains part of the vascular cambium, so it remains undifferentiated. All right, and this type of cell division will enable stems to get wider. Okay, now let's take a look. I'll just clear all that so it doesn't get too messy. Now let's look back here to the left, and you can see something which everyone's familiar with, and these are growth rings, otherwise known as annual rings. And these growth rings or annual rings are actually formed as a result of the different seasonal growth that secondary xylem undergoes. So if you can imagine, to explain how those rings actually form, if you can imagine during a growing season, let's say that this is the outside of the stem. Okay, there. Oh, that's getting messy. Let me try that again. All right, let's say this is the outside of a stem. And we have some, the beginning of the growth season, so growth is very rapid. And the xylem, the secondary xylem cells that form are very large and thin-walled. Okay, so that's how it's growing. All right, then at the end of the growing season, due to environmental conditions such as, decreasing light levels, decreasing nutrients, less water availability and so on, those cells start to become smaller and thicker walled. Okay, there we go. And then at the end of the growing season, growth stops completely. Okay, all right, then we come to the next growing season, so most likely spring, and growth takes off quickly again. And again, we get these large xylem cells being formed. And this basically results in this very obvious contrast, between cells of one season and cells of the next growing season. And that contrast that you can see between the different cell types is what looks like a ring. That is the growth ring or annual ring that you can see there. All right, so hopefully that makes sense to you, and I'll just clear all that and go back down there. All right, and we have a question. Does each layer require 1 year to grow? In theory, yes, that's the case. But in reality, often there's other factors that will slow down the growth and speed the growth up again, even within one growing season. And that's why growth rings, they are just a guide. It varies, yes, it does. They're used as a very rough guide as to the age of a tree. But you certainly can't just say each growth ring definitely equals one season of growth. All right, so let's recap our vascular cambium. So remember our meristem that's producing, let's go back, it's producing secondary phloem which we can see here. This is the secondary phloem that's being produced to the outside of the vascular cambium. And towards the inside of the stem, it's producing secondary xylem. And this is all secondary xylem here towards the inside of the stem. Okay, all arising from this vascular cambium here. All right, so the vascular cambium is a multi-layered cylinder of meristem cells. We just looked at 3 layers of vascular cambium cells. It lies between the cortex, the outer cortex, and the inner pith of the stem, and it's responsible for producing secondary vascular tissues. For example, the secondary xylem and secondary phloem. All right, now let's look at the other meristem, and this is called the cork cambium. So these are meristematic cells that generate the outer covering of the stem, otherwise called the periderm, and the periderm will replace the outer epidermis cells in older stems. All right, so let's look at the cambium. So here's the cork cambium. So these cell layers here, about 3 cell layers once again, and they divide in much the same way as the vascular cambium. So once again, we will have, if we look at this cell here of the cork cambium, they will divide in this direction. So we'll just split that one cell into 2, and this cell will produce a cork cell, and this cell here will remain part of the cork cambium. It remains undifferentiated. All right, in a similar fashion, so this cell of the cork cambium will divide in 2 in that fashion. This inner cell here remains part of the cambium. This outer cell will differentiate to form a thin-walled parenchyma cell towards the inside. And of course, you can see these cork cells here to the outside. They are, of course, dead cells at maturity. All right, and now we can appreciate the different layers of a woody stem. And in fact, we're looking at a tree trunk here. All right, so let's have a look at the different layers, starting from the outside, and we can see the cork is the outer bark. To the inside of that, in green, we have the cork cambium. So remember, these are the meristematic cells. To the inside of the cork cambium, we have the phloem, also referred to as the inner bark. To the inside of the phloem, we have the vascular cambium. That's our second meristem that we're looking at here. To the inside of the vascular cambium, this region here, which is called sapwood. And sapwood actually consists of functional xylem. Functional xylem, sapwood. And then the center of the actual trunk, this is called heartwood. And heartwood was previously functional xylem, but now the xylem vessels have all been impregnated with oils and resins and so on, and it's no longer functional. All right, so let me clear all that and move on. Okay, special functions of stems. So not all plant stems are aerial, as you probably know. There are underground stems, which may be horizontal rhizomes, and from these rhizomes, depending on the plant species, branches may arise or leaves, or they could serve as food storage, as in the potato tuber. So that might be a new one for some students. We always think of potato tubers as being part of the roots, but they're actually underground stems, specialised for storage. Desert plants also tend to have specialised stems. So in arid and semi-arid regions, leaves tend to be absent, or at least significantly reduced to prevent water loss. And instead, photosynthesis is carried out primarily by the stem itself. So, for example, leaves of she oaks are reduced to very small scale-like structures at the nodes of stems. Stems of marsh and aquatic plants are also highly specialised because these environments are usually waterlogged, and even in saline habitats, you might find that with some plant species, the leaves are greatly reduced and the fleshy stems instead take over the primary role of photosynthesis as well as storing water. All right, and here we can see stems of the salt marsh plant, Sarcocornia, and you can see, I'm not sure if you can see, but the leaves are barely visible. They're basically highly reduced, and it's essentially the stems that are performing the function of photosynthesis. Moving on now to our next major plant organ, the roots. So roots, of course, are underground organs, or usually underground organs of vascular plants, and they grow from the apical meristems at their tips, so the root apical meristems. Roots have many functions, of course, including nutrient and water uptake from the soil solution, anchorage and support. There are some very important plant hormones that are synthesized in the roots, and roots also function to store nutritional reserves. Root systems may be branched, which is a common trait of dicotyledonous plants, or they may consist entirely of what are called adventitious roots, which is a trait more commonly found in the monocot species. So, for example, the grasses. And let's have a look at that on the next slide. All right, so dicots tend to have these branched root systems here that you can see. Often there is a central taproot or primary root, and then all the other roots basically arise as lateral roots from the primary roots. Okay, so that's what you commonly find in dicots. But in monocots, you're more likely to find a fibrous root system, which is made up of what are called adventitious roots. And you can see from this image here that the adventitious roots are roots which actually arise primarily from the base of the stem. So you don't have that primary root with the lateral roots or root branches arising from the primary root or primary roots. So that's our adventitious roots in monocots. So let's have a look at root growth, or rather the anatomy of roots a little bit more closely. All right, can I move this up? Oh, here we go. Oh, a little bit too far. All right, I'll just leave it there. All right, so a root consists of an outer epidermis, and we can clearly see that this outer layer of cells, just a single layer of epidermal cells. The epidermis encloses cells of the cortex. So we've got this well-developed cortex here, and then we have one single vascular cylinder, a core or central vascular cylinder, which consists of both the xylem and the phloem. But let's look at the different regions of the root. So firstly, these cells down here, these are cells of the root cap. the term there, root cap. Okay, so these are specialised cells that secrete a substance called mucigel, and that mucigel basically acts as a lubricant to guide and protect the root tip as it's moving between soil particles. All right, behind the root cap cells, we have this region here, which is the root apical meristem. Okay, there it is, the apical meristem, or the meristematic region. Then behind that, we have a region which is called the zone of cell elongation. So these are newly formed cells that have just formed from the apical meristematic cells. And they haven't differentiated yet, but they are still growing, they're still elongating, and they're about to start differentiating. Behind the zone of cell elongation, then we have this region here, which is called the zone of cell differentiation. And of course, you can see that there's a lot more going on here now. So the epidermal cells are very clearly defined. Some of these epidermal cells have elongated to produce what are called root hairs. Don't confuse root hairs with lateral roots, which are quite different. Root hairs are just elongated epidermal cells. Then we have these well developed now cells of the cortex, there and there, and we have the vascular tissue forming in the centre. Okay. All right, so let's take a section of the root through here and then flip over that section so we can see what's going on with the vascular cylinder. All right, so at the moment, all it consists of is a little bit of protoxylem at 4 points there. Then we can take another slice of tissue through here, and now it's a little bit more developed, the vascular tissue. So we've got 4 regions of xylem developing and 4 points of phloem developing also. There's also a layer of cells developing around this vascular cylinder called the endodermis. Very important layer of cells, the endodermis. And now let's take a slice of tissue through here, through the completely differentiated part of the root, and have a closer look. And now we can see the 4 points of phloem that are well developed, and the xylem is completely developed and it looks like a star. Okay, roughly like a star. So just remember that, that the xylem appears as a star shape. All right. Let's recap. So the root cap, so remember the cells right at the very tip, they comprise large parenchyma cells. They're constantly developing, constantly being developed from meristem cells. They secrete a polysaccharide substance called mucigel. And this mucigel contains a mucilage type matrix and also sloughed off living root cap cells. The root surface area is supplemented by numerous fine root hairs. Remember, these are just extensions of epidermal cells, and they're found just behind the growing tip of the root. The soil region around the root hair zone is called the rhizosphere, and this is where it's thought that most interactions occur between the plant and its soil environment, in particular with both free-living and symbiotic microorganisms. All right, so remember the star-shaped xylem that we saw form within the vascular bundle? That xylem is referred to as exarch xylem, and it forms from the outside of that vascular cylinder to the inside of the root. And we have a question. Is there a way to consistently differentiate? Oh, hold on, let me pull that chat box out so I can see it. Is there a way to consistently differentiate between root hairs and lateral roots? That's a very good question. I think that you would find if you looked at a root tip, if you took a root tip and looked at that under just a normal compound light microscope in the lab, you would very, very quickly see the difference between them. The root hairs are really, they're tiny, very, very tiny, very, very hard to see, but the lateral roots are actually very easy to spot. So purely based on size, yeah, you could easily differentiate them. If you went one step further and you cut a section, a cross section through the root tip, and back a little bit, and even a cross section and a longitudinal section, if you could, through the actual root tip, then they're really easy to differentiate. Because you see with the lateral roots, you see the vascular tissue go through the actual lateral roots, but of course that doesn't happen with the root hairs themselves. So quite easy to pick. All right, let's look at the roots a little bit more closely and look at some really interesting features. And I'll just enlarge this for you. Okay, maybe a little bit bigger. All right. So hopefully you can see that all right. All right, so let's look at this again. Okay, this is a rather nice image, this one. So once again, we've got our root cap cells at the bottom that secrete mucilage. We've got our meristematic zone here, so our root apical meristem. But what's interesting is this region of cells here within the meristematic zone. It's called the quiescent center. And this is a group of meristematic cells that don't seem to do anything. They just remain. They don't divide, will very rarely divide. And it's still not, they were identified, you know, a long time ago, and it's still not really clear as to what the quiescent center actually does. The rest of the cells in the meristematic zone, these are madly dividing. Here we have the maximum rate of cell division. And then, of course, we've got that region, the zone of elongation, and you can see already some cells are starting to differentiate here. Epidermal cells becoming very clearly defined, and then the maturation zone there. But that's not what I wanted to point out. I wanted to show you, all right, so let's start up here. So the red cells in the center represent the mature vessel elements of the xylem. Then in green, so this layer of cells in green on both sides, these are cells of the phloem. Here we go, sieve tube elements of the phloem. And then to the outside of that, so this single layer of cells in yellow that you can see to the outside of the vascular tissue, these are called pericycle cells. And they're really interesting because the odd pericycle cell will just suddenly start dividing. And here's an example here. So you can see a massive small cells here. These have actually formed from one pericycle cell, and they'll just keep dividing and pushing outwards. Here it is here until you have an emerging lateral root. So these are how root branches, otherwise known as lateral roots, form. They form from pericycle cells. When they emerge, they actually crush those outer cells as it emerges from the side of the root. So you can clearly see here how different the lateral root is compared to a root hair, which is simply an epidermal cell. a very long epidermal cell. Okay, so that's the pericycle. I think that's all I wanted to show you there. Oh, one more thing too. So that's the pericycle, that's the yellow layer of cells, but to the outside of that, there's another single layer of cells that you can see that are coloured brown. And we're going to talk about that layer of cells in just a minute, and they're called endodermal cells. Endodermal cells of the endodermis. So remember earlier on in this lecture, I said there's a couple of very unique types of dermal tissue. This is one of them. So it's not found on the surface of the root or the surface of the plant at all. It's actually deep within the tissues of the root. So endodermis. All right, so I'll just clear that. And get rid of that. And we have a question, do lateral roots emerge between equal distance from each other? That's an interesting question. Yes, they do. They actually do seem to emerge at sort of specified points. And there's been a lot of work done to actually understand how that happens. How is it that only specific cells of the pericycle actually start to divide? So what are the signals? What are the mechanisms which identify which of those cells will actually start that cell division process to form a lateral root? All right, so let me get that down. So now we're going to look at that cell layer just to the outside of the pericycle, and it's called the endodermis. So outside the pericycle lies a single layer of cells, the endodermis, and these are highly specialized cells. Because the radial walls of these cells are impregnated with a substance called suberin. And this is a lipid type substance which is impermeable to water. So let's have a look firstly at where they are, where these cells are. So once again, so we've got part of a section of a root here, outer epidermal cells. A couple of them have elongated to form root hairs. So most of the root tissue is taken up by these cells of the cortex. Then we come to the central vascular cylinder. So to the inside of the vascular cylinder, we have that star-shaped xylem. And then in blue, we have the 4 points of the phloem tissue. To the outside of the vascular tissue, we have these yellow cells, a single layer of yellow cells. These are cells of the pericycle. So remember, they're the ones that we said give rise to lateral roots. And then to the outside of the pericycle, we have another single layer of cells, which you can see are partly coloured in red. And these are the cells of the endodermis. So let's expand 3 of these endodermal cells and look at them up here. And you can see that in red, this is representing where the radial walls are impregnated with that substance called suberin. In other words, you can imagine anything trying to pass in between those cells would not be able to get through. Instead, it would have to travel somehow through the cells. through the cytosol of the cells rather than in between the cells. All right, let's look at that in a little bit more detail. All right, and to understand why the endodermis functions in that way, let's look at the pathways, the different pathways which water takes to actually enter the root cells. And let's start here with a pathway called the apoplast pathway or apoplastic pathway. And we can see this by following the green line. So this is showing that water is actually moving not through the cells, but in between the cells. In other words, it's actually water's moving basically along the cell walls, following the pathway of the cell walls and in between the cell walls and the cell membranes, but not actually moving into the actual cytosol of the cells. Okay, so this is called the apoplast pathway, and it's moving towards the vascular tissue. Then we have what's called the symplastic or symplast pathway. And in this pathway, water actually moves into the cytosol of the cell. And you can see if we follow the, follow the blue line, you can see that water is moving through each cell, through the cytosol of each cell until it actually reaches the endodermis. And one more, transmembrane pathway. Once again, let's follow this line, but this time we start following the black line. Water is moving through the cells, but it's actually following the route of the plasmodesmata that joins one cell to the next until it hits the endodermis. All right, but what's interesting, once any of these pathways hits the endodermis, let me just clear everything there. And I'll just enlarge that. Hopefully you can see that a bit better. All right, so what's interesting is that once the actual water hits the endodermis, it can no longer follow this apoplast pathway that is moving just along through the cell walls. The water is then forced to take the symplastic pathway, that is, water has to move actually through the cytosol of the cell, which it does so here when you follow the blue line, and it does so here following the black line. So that layer of suberin surrounding the endodermis, which by the way is called the Casparian strip, actually forces water to pass through the symplastic pathway, and this enables the plant to control the movement of water and ions, because it needs to keep water and ions moving towards the actual the vascular cylinder. It prevents the backward flow or loss of ions from the vascular tissue, and it also prevents the loss of sucrose from the vascular tissue back into the cortex of the roots. All right, so very important cell layer, the endodermis and its Casparian strip. All right, how are we going for time? Oh, not too bad. All right, I think it's time for a break. Okay, who's ready for a break? Okay, we're doing quite well for time. Okay, you're ready? All right, so shall we just break until 8 o'clock? All right, I'll see you back here at 8. Right, so just moving on now, very briefly, some special functions of roots, and looking at some mangrove species here. They often possess these upright aerial roots, which we can see here, are called pneumatophores, that function primarily in gas exchange. And that's in addition to these very large prop roots that you can see there. What about roots and saline soils? So some plants, such as river red gums, which are mildly salt tolerant, have one of these unique types of dermal tissue that we mentioned at the start of the lecture. And this is a layer of cells called exodermis. And the exodermis cells are found just below the outer epidermal cells. It's a suberized cell layer, just like those endodermal cells that we saw in the roots, but these ones act as a barrier to toxic chloride ions. And then, of course, we have plants that have storage roots. Storage roots are adapted primarily for the purpose of storage, and they often have additional layers of vascular cambia together with parenchyma cells. So, for example, this is found in sweet potato and beetroot. Moving on now to our final major plant organ, the leaf. And I'll just make that a little bit smaller. Right, so leaves are our major photosynthetic organs. They have limited lifespans. So in some plant species, of course, they're replaced every year. In other plant species, they're replaced every 2, 3 or years or even longer. A leaf usually consists of upper epidermis and lower epidermis, and the epidermal cells will enclose mesophyll tissue, and mesophyll cells are those cells that contain chloroplasts, organelles that you know are called chloroplasts, and they are the ones responsible for photosynthesis. The epidermis of a leaf is covered by a thick cuticle. Well, the thickness of the cuticle really depends on environmental conditions and the plant species. The epidermis, or the cuticle rather, is waterproof. It is impermeable, mostly impermeable to gases. So, of course, in order for gas exchange to take place, the epidermis is studded with these tiny pores called stomata. And those pores can open and close. They regulate water loss from the plant, and they regulate gas exchange between the plant and the atmosphere. And we've got a nice image here of one stomata, or I should say one stoma. Plural is stomata. So we can see in the centre the pore, and this is only just slightly open, this pore here. It is surrounded by two, there's one and there's two specialised epidermal cells, which are called guard cells. And you can see here that guard cells actually contain lots of chloroplasts. So these are unique amongst epidermal cells. Most epidermal cells do not photosynthesize. They lack chloroplasts. That's not their primary function. But guard cells that surround stomatal pores, they actually do photosynthesize. But the primary function of these guard cells is to regulate the size of the pore. They actually function to completely close the pore or completely open the pore, and anywhere in between, depending on environmental conditions. All right, here we have a section of a young leaf on the left here of Eucalyptus globulus, and we can clearly see the different cell types within the leaf. So let's start at the top here, a single layer of upper epidermis, and these epidermal cells secrete that cuticle layer. At the bottom, we have a single layer of lower epidermis, and we can see here and there 2 stomata, and here 3 stomatal pores there. All right, if we look at the mesophyll cells within the actual leaf, we can see that there are 2 major types of cells. So these ones up here, they're closely packed together, these very long mesophyll cells. They're called palisade mesophyll cells. They're all photosynthetic cells. And then the lower part of the leaf contains another type of mesophyll cell called spongy mesophyll cells. They are also photosynthetic cells, but you can see they are quite irregular in shape. They're not closely packed together, so there's lots of large air spaces in between. And obviously that's important because of the gas exchange that takes place via the stomatal pores. All right, to the right here, we've got a mature leaf, and we can see much the same, upper epidermis, lower epidermis, but here we can also see, not very clearly, but we can see a vein. So this is a vascular bundle comprising both xylem and phloem. Once again, we can see those closely packed upper palisade mesophyll cells, and then the lower spongy mesophyll, although they're not as clear in this particular section. Okay, so some of the leaf epidermal cells, depending on species, can also be quite unique. They can have very unique functions. So some of these cells will actually form leaf hairs. Some will actually specialise as secretory gland cells. And of course, those gland cells will accumulate various oils and resins, which are supposed to deter insects that are feeding on leaves. These are interesting crystal-containing cells. So this is particularly evident in some of the grasses, which will accumulate silicate crystals in their cell walls, and that sort of provides a very sharp knife edge to the actual grass leaf blade, and that supposedly deters animals that are feeding on the plants. Okay, so they're just some examples of unique epidermal cells in the leaf. All right, we saw mesophyll tissue. This is the ground tissue of leaves, and mesophyll tissue comprises cells that contain chloroplasts, so the major sites of photosynthesis for the plant. Mesophyll tissue is often differentiated into those palisade mesophyll cells, so the ones that are very closely packed together towards the upper surface of the leaf, and then the more random spongy mesophyll cells towards the lower half of the leaf. Vascular bundles in leaves are referred to as veins, and the veins that you find in flowering plants form this interconnected branching system, with xylem tending to be on the upper side and phloem on the lower side. And in some particular, some plant species, you'll find specialised mesophyll cells called paravenal mesophyll cells. And they are a type of transfer cell. And you might remember we saw an example of a transfer cell earlier in this lecture. And those transfer cells have all those little ingrowths of the cell wall to increase the surface area. And these particular type of transfer cells here form this connection between the phloem and the cells within the leaf. They primarily rapidly transfer sugars. And finally, some modifications of leaf structure. So there's actually some aspects of leaf morphology that's influenced completely by the environment. So you all be familiar with the fact that shaded leaves tend to be larger than leaves that grow in full sun. Shaded leaves also tend to have a very, very thin cuticle layer and compared to leaves that are grown in full sun. But also they're structurally different in the type of mesophyll cells, those photosynthetic mesophyll cells that they contain. Plants growing in particular habitats can also exhibit distinctive leaf adaptations. So, for example, arid and semi-arid plant taxa often have very small scale-like leaves to basically minimise water loss. They have very thick cuticles and lots of leaf hairs. Some plants even possess these very odd pore-like structures, normally at the very, not at the just at the tip, but around the actual margin of the leaf. These very highly modified, they're like stomata, highly modified pores like stomata, but they're open all the time. They don't open and close like stomata. They are called hydothodes. And they actually enable water to keep moving up through the plant, even when the plant is not transpiring, so it doesn't have its stomata open, but it's under what's called root pressure, so the water keeps moving up through the plant. And that extrusion of water through these hydothode pores is called guttation, and it tends to be really common in rainforest plants. And it's also very commonly seen very early in the morning under conditions of high humidity. So that completes our lecture on plant structure. So hopefully you've gained some insight into general plant anatomy. And now we're going to introduce our next lecture. So we're going back now to the topic of DNA. We're going to look at DNA a little bit more closely, and we're going to try to understand basically how information flows. Genetic information flows. But first we need to revise some of those important and often complex concepts that we've already visited in previous lectures. So let's start with some revision. Nucleic acids. You know there's two major types, RNA and DNA, and they are found in all living cells. DNA, of course, is the type of nucleic acid in which information on cell structure and function, and therefore the identity of the organism, is stored and transferred from one generation to the next. So the DNA and the functions of DNA basically enable a plant to be a plant and a horse to be a horse. But how does that happen? That is the question. All right, we know the building unit of DNA and RNA, in fact, is the nucleotide. The nucleotide has three parts: a nitrogenous base, well, one of 5 different nitrogenous bases. It has a monosaccharide, which is either ribose or deoxyribose, depending on whether that sugar is found in DNA or RNA, and it has a single phosphate group. So let's recap. You've seen this slide before. So here we have the general structure of a nucleotide, nitrogenous base, a sugar, and a phosphate. And here it is in more detail. Nitrogenous base, in this case it's an adenine or simply A, and that is bonded to our Our sugar in this case, it's a ribose sugar. So we know this nucleotide is actually found in RNA. This sugar was a 2-deoxyribose, then we know it would be found in DNA. And the sugar is attached to a single phosphate group. I know everyone's very familiar with this because you would have revised it in part for your mid-semester test. All right, the nitrogenous bases are of two types, the pyrimidines, which are the bases comprising just a single ring of carbon and nitrogen. And there are three pyrimidines, uracil, cytosine, and thymine, otherwise known as U, C, and T. And we can see them here. It's uracil, cytosine, and thymine. And then we have the purines. These are the two bases that comprise a two-ring structure of carbon and nitrogen. And here they are to the right. So we've got adenine or A and guanine or G. All right, let's compare DNA and RNA. In DNA, the sugar, the monosaccharide, is called a 2-deoxyribose. And this means that the hydroxyl, that is the OH, at the 2' position of the sugar is replaced by a hydrogen. And the bases found in DNA nucleotides are A, T, G, and C. So let's go back just to make sure that you're following that. Here we go. All right, so if we were looking at a nucleotide that was found in DNA, this sugar wouldn't be a ribose, it would be a 2-deoxyribose, which means that at the 2' position, which is here, this hydroxyl group, this OH, would not exist. Instead, there would simply be a hydrogen there, a single hydrogen at the 2' position. And that's basically the difference between a ribose and a 2-deoxyribose. Okay, so RNA, of course, has a ribose sugar, and the bases are adenine, uracil, guanine, and cytosine. Now, DNA, as you know, exists as two long chains of nucleotides running in opposite directions and wound together in a double helix formation. RNA, on the other hand, always exists as a single chain of nucleotides, just a single chain of nucleotides. So very different in that respect. RNA is also a very transient molecule. It is broken down very quickly after it's synthesized, whereas DNA is very robust, it's very long-lived. All right, structure of DNA, you've also seen this slide before. So here is the representation of a DNA double helix. Here's the ribbon, a ribbon structure. Also a double helix. This one's a little bit easier to follow. So firstly, let's look at the two chains of DNA. So we'll look at the pink ribbon first. This is the 5' end. And up here we have the 3' end. So what are we looking at here? What is the ribbon actually representing? The ribbon represents what's called the backbone of the DNA. And the ribbon consists of the sugars and the phosphates of the nucleotides. So that's referred to as the sugar phosphate backbone. And that's what we're seeing here within the ribbon. Okay, and the same thing for the other strand of DNA, which of course is 3' here and 5' here, because they are said to be anti-parallel. All right, so I'll just clear that. All right, and then towards the inside of this DNA double helix, we can see the actual bases, the nitrogenous bases found towards the inside, and they are, of course, bonded together via hydrogen bonds. All right, so let's take just this region here and expand it to what we see here on the right. And of course, we can see two nucleotides here. They're on one strand of DNA and two nucleotides here on the other strand of DNA. And you can see that these two here are upside down relative to these nucleotides on the opposite strand. All right, very important now are the ends of the DNA. So this is the 5' end of this strand of DNA. So we call this the 5' phosphate. 5' phosphate. At this end of the DNA, we have the 3' end, and we refer to the 3' hydroxyl on this sugar. That's what the 3' is referring to. It's referring to the hydroxyl group, the OH group on this particular sugar here. All right, the same thing applies to the opposite strand of DNA. Here we have the 3' hydroxyl of this sugar, and this end of this strand of DNA is referred to the 5' end because we're referring to this 5' phosphate group. And then, of course, you can see the hydrogen bonds that are linking those two chains of DNA together. All right, we can also see here that a cytosine always binds to a guanine on the opposite strand, and a thymine on one strand of DNA always bonds to an adenine on the opposite strand of DNA. So that's when we're looking at two strands of DNA. But what if we were looking at, a strand of DNA and we had a cytosine and we had a thymine, and here we've got an adenine and a guanine. All right, so this is DNA, one strand of DNA. And let's say we had here a strand of RNA. So here's the backbone. All right, so what would be the nucleotide here in the RNA strand that would bond to a cytosine in the DNA? You can tell me. So what we're looking at here is referred to as the base pairing rules, G, correct? Yep, so just like in DNA and DNA, a C bonds to a G. What about the next one? So we've got a thymine in the DNA. That would bond to what nucleotide in the RNA? An adenine, correct? Yep. RNA. And let's go to the bottom one. What would bond, if we had a guanine in the DNA, what would we have in the RNA? What would bond to it in RNA? Yep, cytosine. All right, and here's the tricky one. This is where we need to know the difference between the nucleotides in DNA and RNA. So we've got an adenine in the DNA. Yes, that's right, uracil. So rather than having a T here, which is what we would have if it was DNA to DNA, remember RNA lacks thymine, and instead of thymine, it uses uracil to bond to adenine. Okay, good. So these are referred to as the base pairing rules, and you should be familiar with both the base pairing rules in DNA, from DNA to DNA, and from DNA to RNA. All right, and this will set you up well for one of our lectures next week. All right, so I believe I've covered everything here, so let's move on. Let's talk about chromosomes. So DNA is organised into chromosomes. Each chromosome is a single DNA molecule. In other words, two strands of DNA bonded together makes up a single molecule of DNA. In eukaryotic cells, so in plants, in animals, in fungi, in protists, chromosomes are located in the nucleus and they are characteristically linear chromosomes. In other words, if you stretched out the DNA, oh, that is a very poor DNA double helix. There would be distinct ends to the DNA. They are linear chromosomes. In comparison to prokaryotes, which often have circular chromosomes. They're the two strands of DNA, and we have one circular chromosome. All right, each species of organism has a unique chromosome complement. That is unique chromosome shapes, sizes of the chromosomes, and number of chromosomes. Just a couple of terms here. So a region called the centromere is located in the middle of the chromosome. It's essential for cell division. And the two ends of each chromosome are called telomeres. So these regions here are called telomeres, these regions called telomeres, and there's a region sometimes in the middle, sometimes offset a little bit, which is called a centromere, and that's important for cell division. All that. All right, here we have a very interesting image. This is called a karyotype. A karyotype is simply a collection of all the chromosomes that you would find in the nucleus of one cell, of one single cell. And this is a karyotype of a human cell. Let me just enlarge this a little bit more. Okay, that's better. All right, so let's start here. So we've got a karyotype from a male human, and we can see the total number of chromosomes here in the nucleus of a cell from a male human is 46 chromosomes, 46 in total. That 46 consists of 22 pairs of chromosomes plus 2 additional chromosomes. The 22 pairs of chromosomes that you can see here, and by the way, these are real chromosomes. This is an image of actual chromosomes that have been isolated from a cell nucleus. So this is what they actually look like. And someone has painstakingly separated these out, ordered them, and lined them up really well without breaking any of them. So really well done. Very well done. Not an easy task. All right, so the 22 pairs of chromosomes that you can see here, those 22 pairs, they are called autosomes. Autosomes. And these two extras here are called sex chromosomes. So the X and the Y are called sex chromosomes. And of course, they determine gender. Sex chromosomes. I'll just write there. that abbreviation there. All right, so you can see 2 copies of chromosome number 1, 2 copies of chromosome number 2, and so on and so on. All right, so what's the significance of these pairs? So if we look at these 2 copies of chromosome number 1, they are referred to as homologous chromosomes. Homologous. Homologous chromosomes. And what this actually means is that these 2 chromosomes are very similar, but they're not exactly the same. They're very similar in that they carry, they consist of the same types of genes, but those genes might not be exactly the same. They're the same genes in terms of the trait that they control, but they might be a little bit different from each other. So just as a really simple example, let's say, and this is not the case, but let's say the gene that controls eye colour, your eye colour, is found on chromosomes number 1. And let's say the gene is found here and here. Okay, so they're both genes that control eye colour, but this one might be for brown eyes and this one might be for blue eyes. So now you can understand why you've got the same type of gene, but they may be a little bit different from each other. All right, so there, that's basically what we mean by homologous chromosomes. All right, let me just clear that. All right, the sex chromosomes. So males have one X and one Y chromosome, and these, of course, determine maleness. On the other hand, if we look at a karyotype for a female, just scroll down a bit. Here's the karyotype for a female. Oh, gone down a little bit too far. So if we look at the sex chromosomes for a female, there are 2 X chromosomes. Obviously, there was a problem straightening out these chromosomes here, as you can see, but overall, a really good job. All right, there is a problem with this particular karyotype in that you can see down here, there are 3 copies of chromosome number 21. And this unfortunately causes the condition called Down syndrome. So rather than 46 chromosomes, which would be normal, this individual has 47. This is also known as trisomy 21. And having too much DNA or not enough DNA can cause enormous problems. So in this case of Down syndrome, the individual is prone to heart conditions, intellectual disability, and a whole range of other problems. And there are lots of other trisomies, not just trisomy 21, but other trisomies actually cause babies to be stillborn, or they die soon after birth, or they don't even survive full term. All right, so that's the karyotype for a female. All right, an important point to keep in mind is that all these chromosomes are found in a single nucleus of most cells in the body of the male. Most cells. And the cells that contain all of these chromosomes are called somatic cells. Somatic cells. And we're talking about most of the cells. Somatic cells, 46 chromosomes. Okay. Then you have some cells that contain only 1 copy of each pair, only 1 of each pair. And these are the reproductive cells. Reproductive cells. reproductive cells contain only half the number of chromosomes, in other words, 23. And what are the reproductive cells? We're talking about sperm cells and, of course, egg cells in females. Sperm and eggs. Right, so I think we've covered everything that we need to cover for now. Clear that, and we can move on. All right, so chromosomes in eukaryotes are made up primarily of DNA, which you're very familiar with now, and that DNA is associated with various proteins. Proteins that associate with DNA are called histones, and they assemble in groups of 8 and form a core of proteins upon which the DNA is bound. And together, this is called a nucleosome core particle. So let's have a look at that, I believe, on the next slide. Yes, so here's a nucleosome that we can see. So in blue, we have the double-stranded DNA, and it's wound around a core of histone proteins, and they're given funny names like H4 and H3 and so on. So there's the histone proteins. Here's another histone protein, H1. Okay, and then there's a little bit of DNA that's not wound around the histones, and it's called linker DNA. And if we were to follow this DNA along, you would find that the next region of DNA would wind 1.5 times around another core of histones. So that's basically what a nucleosome looks like. All right, so now we can actually understand basically what goes into what we normally see as a chromosome in various images. Actually, take note, this is not just one chromosome here, this is 2 chromosomes joined together at this region here, the centromere. All right, so here's our double-stranded DNA. Let's follow it round. Here the DNA starts winding around those histone proteins. Okay, the nucleosomes now wind even further, and you can see how condensed the DNA becomes as it forms an actual linear chromosome. So this is one chromosome here, and the chromosome has replicated to form a copy of itself. There we go. They're linked together. They're still joined together. They haven't separated yet. All right, we're nearly there. Okay. All right, so we're going to finish with this classic experiment that was conducted in the 1940s by 3 individuals, Avery, McLeod and McCarty. And you have to imagine that at the time they knew about the structure of DNA. They'd identified the nucleotides, the chemical composition of the nucleotides. They knew that DNA was associated with proteins. What they didn't know was which one of those molecules actually carried the genetic information. Most people at the time thought that DNA was too simple. It was too basic. It only consists of 4 different nucleotides. How can that possibly carry genetic information from one generation to the next? So most people believed that it was the proteins that were associated with the DNA, because there are so many different types of proteins found with the chromosomes. All right, so in 1944, these chaps actually demonstrated that it's actually the DNA, not the protein, that carries the genetic information. They did this by observing two different forms of the same bacteria. And the bacteria is called Streptococcus pneumoniae. It causes pneumonia, meningitis, septicemia, and lots of other infections. All right, they had two different strains of Streptococcus pneumoniae. One was virulent, that means disease causing, and they called it S for the smooth strain. The other strain was not virulent, didn't cause any problems, and they called it R for rough strain. So let's look at what they did. Here is the experiment. They took the rough, non-virulent strain of bacteria, injected that into mice, and the mice were fine. They remained healthy. If they took the smooth, virulent strain, the S strain, injected that into mice, the mice died. Then they took the virulent, the smooth, virulent strain, but they exposed it to enough heat to actually kill the bacterial cells. So heat-killed, virulent strain, injected those into mice, and the mice were fine, remained healthy. But here's the interesting part. They took the non-virulent strain R bacteria, mixed those with the heat-killed smooth virulent strain, mixed them together, injected those into mice, and the mice died. So why would that happen? So the heat killed on its own, no problem. Virulent strain on its own, no problem. But you put them together and the mice die. Okay, to understand this, we need to know a little bit more about certain bacteria, certain species of bacteria. Certain species of bacteria, so there's one bacterial cell, are said to be competent. Competent. That is, they can actually take up naked DNA, bits of DNA from the environment. That is, they are said to be transformed. They are transformed. So when these cells were actually killed, you'll have, broken bits of cells, cells broken open, and then you'll have little bits of DNA that have actually come out of the cell, little bits of DNA. And those little bits of DNA can be taken up by live bacteria, certain species of live bacteria. So in this case, these non-virulent bacterial cells actually took up some of the DNA, that is, they were transformed with some of the DNA from the virulent bacteria, and they just so happened to take up the DNA that confers virulence. The DNA that contains the genes that confers virulence and causes disease. And that's why when you put them together and inject those into mice, the mice actually died. So this basically showed that it's the DNA component, because these competent cells, they are not transformed with proteins, they are only transformed with DNA itself. And remember, the heat killing actually destroys the proteins, denatures the proteins because of the high heat that's used to kill these cells. All right, so let's go back now and we can understand this process here. So mice injected with live non-virulent bacteria and heat-killed virulent bacteria died. Neither preparation on its own killed the mice. The non-virulent strain was said to be transformed by the virulent material. That is, the non-virulent strain of bacteria could actually take up bits of DNA from the environment. The virulence acquired from that transformation process was even passed on to the progeny of that transformed bacteria. And that's it for tonight. Thank you, everyone. That completes our lectures for tonight. Thank you, everyone, for your wonderful attention. I'll be staying on for a little bit in case anyone wants to chat about anything. Anyone's got questions? I'll just stop. I'll stop the recording here. Oh, actually, no, I'll cancel that. I won't stop the recording because I still need to go through a couple of things. All right, so here's the tutorial just based on the structure of plants lecture tonight. These questions are not assessed, but of course they're still very, very important to cover at some stage before the final exams. All right, moving on here. I think everyone knows that prac report due dates are coming up. So of course, if you haven't already submitted prac report number 1, oh, where are they? Oh, here we go. If you haven't already submitted prac report number 1, don't forget to submit by the 15th, so that's this Friday. And then for everyone, prac report number 2 is due on the 29th, so the last day of semester, the last Friday of semester. All right, so that's the prac reports, and let's go to tutorial questions. So there is one more set of tutorial questions assessed, and they will be based on next week's lectures. Well, we've already covered half of one tonight. We'll finish that next week, and then we'll also complete this lecture on the genetic code. Okay, so that's your assessments. Okay. Okay, I think everyone knows what pracs they need to be writing up. Okay, so that one there, standard curve for prac report number 1, and acid-base titration for prac report number 2. And finally, exam schedule. So not quite completed. I think I'm just waiting on, all right, so that's confirmed, that's confirmed, that's confirmed. So is that one. Okay, this one, equine locomotion exercise fees. I just need to confirm whether that's going to be a split exam or not. That one's confirmed. This one's confirmed. That one's confirmed. And I'm just waiting on confirmation for ag marketing and commodity trading, which I don't think anyone here is doing. All right. Any questions? All right, now I can stop the recording. All right. Stop.